And I'm going to start recording, and we're going to go to the next set that we're going to look at. And we've been talking about the Hardy-Weinberg equation. Before we start working some problems, we're going to look at things that affect our results on the Hardy-Weinberg equation. So you should be looking at a page that says microevolution and factors that disrupt Hardy-Weinberg equilibrium. If you don't see that page, let me know, and I'll check. I may have the wrong page selected. So if we have a population in Hardy-Weinberg equilibrium, allele frequencies are constant from one generation to the next generation to the next generation. And we can use those allele frequencies to predict genotypes and phenotypes. But if we look at natural populations, if you would go out in your backyard and look at the population of crickets, or if you would go to a nearby lake like Wolf Lake, and you would look at the population of smallmouth bass, a type of fish that's found in that lake, what you would see is that a lot of populations in nature aren't in Hardy-Weinberg equilibrium. Now, you might think that if the Hardy-Weinberg equilibrium doesn't actually apply to all the populations in nature, it's kind of pointless. But actually, when we look at the Hardy-Weinberg equation, we can look at a population and we can treat it as if it were in equilibrium. And we can see what predictions the Hardy-Weinberg equation makes if we know what P and Q are. And then we can see in real life how the predictions of the equation are different from what we observe in the real population. And a lot of times this can give us clues about what's happening in the real population. So to do this, we have to keep in mind that there are 6 major factors that disrupt the Hardy-Weinberg equilibrium. And when I say that they disrupt the Hardy-Weinberg equilibrium, that means that the Hardy-Weinberg equilibrium is not true in that population. So remember, the Hardy-Weinberg equilibrium is based on the idea that P and Q, the frequency of the dominant allele and the recessive allele, are constant from one generation to the next. So things that change allele frequencies are things that disrupt the Hardy-Weinberg equilibrium. So something that we talked about back in 105 but applies in this situation is mutations. If an allele mutates, we could have big A, and after a mutation, we have little a. So we have a dominant allele, and after mutation, we have a recessive allele. So this would change the allele frequency. We have what Darwin talked about in his book, natural selection. And natural selection is sometimes called differential survival, meaning not everybody has the same chance to survive. You know, I think I used zebras as an example last week. Zebras that are fast runners have a better chance to survive than zebras that are slow runners. Zebras with strong leg bones have a better chance to survive than zebras with leg bones that break easily. Gene flow, which is sometimes called migration. Individuals moving into the population are out of the population. Genetic drift, which is sometimes called random genetic drift, because as we'll see in a few moments, it's a random process. There's no way to predict what's going to happen with genetic drift. Genetic recombination. One of the main types of genetic recombination that we talked about back in 105 was a process called crossing over. So down here in the lower right-hand part of the diagram, it shows 2 homologous chromosomes. Remember, we tended to use blue chromosomes for the chromosome an individual inherited from its father, red chromosomes for the chromosome that an individual inherited from a mother. And remember, in crossing over, we get a little section of the chromosome, that little red piece down here, from the mother on the chromosome that mostly has genes from the father, and vice versa. There are other types of recombination too, but for now we'll stick to that one. And another process that's called sexual selection or non-random mating. And sexual selection came about in terms of natural selection. Remember what we said at the beginning of the class. There's really two things that living organisms have to do to survive. They have to be successful. They have to survive, and then they have to reproduce. Well, in Darwin's little book on the origin of species, he mostly talks about natural selection, which individuals survive. After that book was published, Darwin recognized, okay, surviving is nice, but if you can't convince a member of the opposite sex to mate with you, you don't pass any genes down to the next generation. So you don't really contribute to the next generation. So Darwin wrote another book on what he called sexual selection, which is sometimes called non-random mating. And non-random mating probably gives you a better idea of what's involved, but sexual selection, on the other hand, is kind of parallel to natural selection. Natural selection is differential survival. Different variations have different chances to survive. Sexual selection is differential reproduction. Different variations have different chances to reproduce. And as I say, it's sometimes called non-random mating because it's not just picking a name out of a hat and deciding that that's the person you'll get married to and have children with. And in Darwin's book, he actually talked about how sexual selection had most likely had an impact on human evolution. So we're going to look at all these. Tomorrow, whether we finish with all these or not, we'll probably talk about them a little bit more, but we will work some problems, especially involving natural selection and just kind of the basics of the Hardy-Weinberg equation. So remember what I said, make sure that you have some kind of calculator app on your phone or your tablet or computer, whatever you're using, because we will need to do that. And you want to make sure it's a calculator that will take square roots and will square numbers. Other than that, as long as it does the 4 basic operations, you should be good. So let's go down the list kind of one by one and look at these factors that can disrupt Hardy-Weinberg equilibrium. And keep in mind, these are the main factors. There are 2 or 3 other minor factors that can disrupt equilibrium, but they're very rare. So we generally don't worry about them. So let's think about mutations first. We'll go through them in the order that they were listed. So remember that mutations are random changes in the DNA. And in terms of how they affect the amino acids in a protein, there are 3 basic types of mutations. There are silent mutations. Well, if you remember that genetic code chart that we used at the end of Biology 105, and remember what it gave us was a 3-letter codon, which we found on the messenger RNA, and then it told us the amino acid that 3-letter codon stood for. Well, remember that there were some codons that stood for the same amino acid. So for instance, there were 2 for phenylalanine, U, and UA. So if we have a mutation and we end up getting a piece of messenger RNA that says UUA instead of UUU, we still get the amino acid phenylalanine in our protein. So this is called a silent mutation. The DNA is changed, but we still get the same amino acid. So notice if we get the same amino acid, this has no effect on our protein. So that's why this is silent. It's not really something that we can observe. And then remember, we also had some mutations that we called sense mutations. And in a sense mutation, we get a new amino acid instead of the old amino acid after we have a mutation. So we'll look at an example of that in just a moment. And then remember, we also have nonsense mutations. And in a nonsense mutation, we're supposed to get an amino acid, but instead we had one of those stop codons. So mutations happen, and when we say that they're random, we have to keep in mind what we mean when we say random, because it's a lot of different ways that we could mean random in this sense. And what we mean here is when we have a mutation might be beneficial. It could change the protein so that it works better. Or maybe it changes the protein so it does something completely different in metabolism, but it's a good change. Mutations can be deleterious, which means bad. And it used to be thought that most mutations were deleterious. There was often an example given that said, imagine that you have a race car that you're looking at something at the Indy 500. And we're going to give you a couple of tools to work on a car, and we're going to put you in the pit. And when they make a pit stop, you're going to make some adjustments to the engine. Well, what are the odds that car is going to run better after you make some adjustments? Probably not real good. So the argument was living organisms' DNA is so finely tuned, if we change it at all, it's probably going to be bad. But there's a third possibility. Mutations can actually be neutral. They don't really change how the protein works. Now, if you think about it, silent mutations would obviously be neutral. And most biologists today seem to believe that the majority of mutations are probably neutral. But when we say that mutations are random, if there's a mutation, if there's a change in the DNA, you can't predict whether that's going to be beneficial or deleterious or neutral. If you know enough about the chemistry and you see which amino acid gets substituted for another one, you might be able to predict. But just because there's a mutation, you don't really know. Now, because mutations are random, this means they don't necessarily make something called adaptations. We'll talk about adaptations in a little bit, but adaptations are characteristics that help an organism to survive and to reproduce. Now, some adaptations are good for survival, not so good for reproduction. Some adaptations are good for reproduction, not so good for survival. And some are good for both. So adaptations could be part of the anatomy. If you're a zebra, adaptations could be going way back to your ancestors, the zebras that began to develop stripes. Remember, we said that stripes provide camouflage for zebras. But adaptations could be physiology. They could be how living organisms work. So they could be the biochemistry that's going on in the cells. And at least in animals, adaptations could also be behavior. So adaptations are things that help an organism to survive or reproduce, or both. So here's an example of a mutation that we looked at back in 105. So we have some DNA, and the DNA that's on the lower part here is our sense strand. Now remember that the sense strand is what we use to make our messenger RNA. So if our DNA is CTC, we're going to make messenger RNA that's GAG, and this gives us an amino acid called glutamic acid in our protein. The protein is something called hemoglobin. Glutamic acid is a polar amino acid. In the most common mutation for sickle cell anemia, this A is mutated to a T. And then that means that the sense strand becomes CAC. So now we make some messenger RNA, GUG, And if we would look that up on the chart that we used last semester, this gives us an amino acid called valine. And valine is nonpolar. Now, if you would look at a human red blood cell, there are millions of molecules of hemoglobin in the cytoplasm. That's just about all that there is. And if this is a polar amino acid like glutamic acid, all these molecules of hemoglobin kind of float around in the cytoplasm. But when we have the mutation that causes sickle cell, valine is nonpolar, and all the hemoglobin molecules, or most of them anyway, clump together in the red blood cell. And when they clump together, they stretch the red blood cell out of shape. And this is what gives the red blood cell its sickle shape. So this is what causes sickle cell anemia. So this would be an example of a deleterious mutation. Now, notice that mutations are not random in a chemical sense. So for instance, here we see A mutating into T, and mutations depend first of all on whether it's A or C or G or T that's being mutated, but they also depend on the location inside the gene. Certain locations in genes are more likely to mutate than other locations. So we don't want to think that mutations are random in a chemical sense, because they're not random in that sense. They're random in terms of the effect that they have on the protein. Do they make the protein better, worse, or not really impact it? So we want to keep in mind what we mean when we say random. Now, Charles Darwin's idea of natural selection, what we talked about, his basic ideas, this to date is the only satisfactory explanation that biologists have been able to test in an experiment up to this point in time. So Darwin's basic idea of natural selection: not all individuals in the population survive long enough to reproduce. And after Darwin wrote his book in 1859, one of the reasons that his book was successful, where people who came and talked about change before Darwin weren't successful, one of the reasons was because natural selection was a hypothesis that could be tested in an experiment. So in all these experiments on natural selection, there have been three basic types of natural selection that have been discovered. One is directional selection. And in directional selection, individuals that have one extreme value of a trait are more likely to survive, and individuals that are at the other extreme are less likely to survive. Keep in mind, in just a minute, we'll see some examples with some pictures, and these will be a little bit more clear. In stabilizing selection, individuals with both extreme values of a trait are less likely to survive. Individuals who are in the middle, who have a medium value of a trait, are more likely to survive. And in disruptive selection, or it's also called diversifying selection, individuals with the medium value are less likely to survive, and individuals at both extremes are more likely to survive. Sorry. Now notice that when natural selection works, it works on phenotypes. Keep in mind, we're talking about things that change the gene pool, the frequency of the alleles. But if certain phenotypes don't survive when they die, they take their genotypes with them. So let's look at three examples that we see. So here's an example of stabilizing selection. This blue line represents the distribution of how many eggs that robins lay. So over here on the left-hand side is 0, 1, 2, 3, 4, 5, and about the most eggs that robins lay is about 8 or 9. So notice that this distribution, if you would take a statistics class, the original population is sometimes what's called a normal distribution, or it's sometimes called a bell-shaped curve. If you look at the shape of that blue line, it vaguely looks like a bell, I guess. In stabilizing selection, individuals that lay just a few eggs aren't usually successful, because when robins lay their eggs, there's almost always one or two eggs that blow out of the nest on a windy day, or maybe they get eaten by a predator that eats eggs. So they're not real successful in raising young, because if you only lay one egg and it blows out of the nest, that was it. Robins who lay a lot of eggs tend to not be very successful, because even though they might have more eggs hatch, when the eggs hatch, there's too many mouths to feed. Some of the baby birds starve to death. And instead of just a few eggs where everybody gets enough, everybody's malnourished. So you might lay 8 eggs, but only 1 or 2 survive. So for robins, the optimal size, kind of where the peak of this red line is right around 4 eggs. Robins can catch enough food to feed 4 eggs. And if they lay 4 eggs, probably 3 of those, or maybe 4, will survive. If they lay 5 eggs, that might work out. If they lay 3 eggs, that might work out. But if you get down to 2 or 6, it's probably not going to be successful. So this is an example of stabilizing selection. Another example of stabilizing selection that's kind of closer to home, we would see similar results if we looked at birth weight in people. Babies who are extremely light are less likely to survive. Why? Because they're probably born prematurely. Sometimes their organ systems aren't formed. They may not survive. They may have to be on a machine that breathes for them and pumps blood through their body, and they may not live. Babies who are extremely large are less likely to survive. Why? Because if you're extremely large, there can be complications during birth. Normal birth may not be possible, may have to do a C-section. There are some other possibilities. So for people, the optimum birth weight is somewhere around 6 pounds and I think 10 ounces or something the last time I read. Now notice, this is also an example of how human culture can affect natural selection. Imagine that birth weight is something that's partly inherited. So if you're unusually light when you're born, your children are going to be unusually light. Or if you're unusually heavy when you're born, your children are going to be very heavy when they're born. If you go back in history a few hundred years, when very light children and very heavy children, maybe I should say babies instead of children, didn't usually survive birth, then those genes would be reduced in the population. But now with modern technology, we can keep babies who are very light alive. We can keep babies who are very heavy alive. So those genes tend to stay in the population more than they would without medical intervention. Now notice, that doesn't mean that we're stopping evolution, because if the current human weight chart looks like this red line and it's gradually spreading out, that's also evolution of a sort. It's not stabilizing evolution, as we'll see in a moment. So in stabilizing selection, individuals in the middle are more likely to survive. So it's kind of like squeezing our graph from both sides. The graph gets skinnier, but it has a higher peak. Another possibility is directional selection. And for directional selection, what we see is there are some moths called peppered moths. And peppered moths get their name because they have black and white spots on their wings. Some of them are almost completely white wings. Some of them have almost solid black wings. But most peppered moths have gray wings that are somewhere in between. Some have very light gray wings that are almost white. Some have very dark gray wings that are almost black. So on this graph, white wings are over towards the y-axis on the left. The blacker wings are over towards the right end of the graph. And if we have a population where, again, we see that normal distribution to start with, if light-colored moths are less likely to survive, then it's mostly dark-colored moths who survive and have offspring. So what happens in the next generation? It's kind of like pushing this graph over to the right a little bit. And this is actually what happened during the Industrial Revolution in England. Peppered moths in general, are active at the night, and they pretty much are quiet during the day. Peppered moths tend to sit on tree trunks, or hang on tree trunks, maybe I should say, and they kind of blend in with the bark. And the main thing that eats peppered moths, their main predator, is different types of birds. So a moth who blends in with the tree bark will be better camouflaged. A mark that sticks out is going to be easier for predators to see and eat. So before the Industrial Revolution, most tree trunks were a little bit lighter, and lighter colored moths had a better chance to survive. One of the things that happened during the Industrial Revolution was that the first fuel that was burned in most parts of the world, including England, was coal. And coal puts out a lot of soot when it's burned. And that soot is released from the factories that burn it. And the coal soot collects. It falls to the surface of the earth, and a lot of it sticks to trees. So during the Industrial Revolution, when everybody was burning coal for energy, that soot made the tree trunks darker. And as a result, light-colored moths started showing up better to the birds. So birds were able to find and eat the light-colored moths more easily. Dark-colored moths were more likely to survive and reproduce. So the population of moths became darker. Now notice about the 1970s or so, this began to reverse and light-colored moths became more common again. Why? Because during the 1970s, people became a little more environmentally aware, and they put in pollution controls, so there wasn't as much soot being released from factories. So gradually the soot that was on tree trunks washed off in the rain, and now the tree trunks were lighter again. Light-colored moths had a better chance to hide from the predators and survive. Now, the last example, the last diagram that we have here is diversifying selection. And they kind of made up an example with rabbits. I don't know why they didn't use a well-known one with moths again, not peppered moths. In this case, we have a population of rabbits, and some of the rabbits on closer to the y-axis have darker fur. The ones in the middle have white fur, and the ones at this end have mostly light fur, but they have dark fur on the tips. Very similar to if you've ever seen what are called Siamese cats. These rabbits that have the ears and the tips of their paws and their tails are usually dark are called Himalayan rabbits. So we have all these types. In diversifying selection, if you put these rabbits on a rocky environment, the dark-colored rabbits and the rabbits with dark patches have better camouflage. And because they have better camouflage, they're harder to spot. But there aren't very many white rocks in nature. And so as a result, the rabbits with the white color, the ones that are in the middle of the population, are less likely to survive. So notice again, we start out with that normal distribution, and we end up with something that kind of looks like a camel's two humps. And this is because the population is split into two separate groups. Now notice this doesn't necessarily change the average color of a rabbit. We still have just as many on the left as we do on the right. But the difference is, before, most of the rabbits are clustered around the average value. At the end, we have a lot of rabbits that are at one extreme, a lot of rabbits at the other extreme, not that many in the middle. There is a moth that's found in Africa where they range from blue to orange, and then purple is in the middle. And purple is heterozygous. So, you know, the purple color is kind of a mixture of blue and orange. And it turns out that the blue moths resemble another type of moth that's blue and it's poisonous. So birds avoid eating them. And the orange moths resemble another type of moth that's orange and it's also poisonous. So the birds avoid eating it. But the poor moths in the middle that are purple, there aren't any poisonous purple moths in the area. So the birds know they can eat them. So we have a population that's mostly blue moths and mostly orange moths. And there are a few purple ones that are heterozygous, but not very many, because those ones are easy for the birds to, first of all, spot. There's not a lot of purple in nature, but also once they spot them, the birds know they can eat them. So we see three different patterns. Now notice when we look at a population in nature, think of the peppered moths that I mentioned before. Natural selection doesn't necessarily stay the same generation after generation after generation. So peppered moths, if we look at before the Industrial Revolution in the 1600s and early 1700s in England, and the reason we know is because people made insect collections. We can look at some of these insect collections that have survived, and we see that the moths were mostly light. After the Industrial Revolution began and pollution made tree trunks darker, the population of moths gradually shifted until dark moths were more common. After environmental controls were put into place, now it's shifting back to light moths. So one of the things we want to keep in mind about really any of the things we're discussing with these factors that change the Hardy-Weinberg equilibrium, we're changing the allele frequency in the population. So this tends to be a very slow, a very gradual process. And depending on what's happening in the environment, it can reverse directions. It doesn't necessarily have to stay the same always. Because remember, Darwin's idea of natural selection is the local environment, what the conditions are right where that organism is living. The local environment is really what controls who survives and who doesn't. Or maybe I should say who has a better chance for surviving and who has a lower chance for surviving. So the advantage of this, if you think about this from a practical standpoint, is it's very easy to conduct experiments about natural selection. We can collect peppered moths. We can catch some of the birds that eat them, and we can put peppered moths into a controlled environment where we control how dark the tree trunks are, and we can see which moths survive and which moths don't survive. We can climb trees and we can see how many eggs are in a nest after robins lay them, and then we can follow that nest and we can see how many hatchlings emerge and survive. So the nice thing about natural selection, and really what separates Darwin's ideas from the people before him, was he had a hypothesis that could be tested in an experiment. We have to have a hypothesis that can be tested and can be disproven in an experiment, or we don't have a scientific idea. So a lot of the people who talked about evolution, who talked about change before Darwin, their ideas made logical sense, but they didn't really have a hypothesis that could be tested in an experiment. Now, Darwin had a second advantage. Not only did he have a hypothesis that could be tested in an experiment, but when scientists did experiments to test his hypothesis, they couldn't disprove it. So for your ideas to stick around, not only do you have need a hypothesis that can be tested in an experiment, but it has to be a hypothesis that survives that test. If it gets disproven, you got a scientific idea, but it's gone. Gene flow is just called migration. And this is just the movement of individuals from one population to a second population. Emigration refers to individuals that are, oops, I have this backwards actually. Emigration refers to individuals moving out of a population, and immigration refers to individuals moving into a population. Notice that these two events always have to occur together, just from the perspective of two different populations. If you live in Illinois and you move to Indiana, you're immigrating into Indiana, but you're emigrating out of Illinois. So if an individual emigrates out of one population, they need to immigrate into another one. And if you think about individuals coming into a population or leaving, it's going to be very rare for them to have the same allele frequency as the population that they're coming into or going out of. So let's look at an example. Let's think about, instead of an allele, let's just look at X and Y chromosomes for a minute. So let's look at a classroom and let's make it a small classroom. So, oops. So we've got 4 students in our class. We've got 2 males, they're both XX, and we have 2 females, they're both XY. So let's think about the frequency of the X chromosome and the Y chromosome. So notice there are a total of 8 chromosomes here. If we look at the X chromosome, 6 of the 8 chromosomes in our population, or if we want to reduce that and simplify the fraction, 3/4 of them are X. If we look at the Y frequency, the Y frequency is 2/8 or 1/4. Okay, so now let's think what happens. So we have a female that joins this class. I hope if I could type. So a female comes into the class, she's XY. So now let's think about this female. If we just look at this female, this female is, her allele frequency is 1/2 X. Whoops, I said she was a female. How about if we make her XX? She's 100% X. Our population isn't 100% X. So when she comes in, this is going to change the allele frequencies. Similarly, if we have one of our females, this one, leaves. If an individual enters our population or leaves our population, it's very unlikely they're going to have the same allele frequencies that we see in the population as a whole. So this means when they come in or when they leave, they change our population allele frequency. So notice that if we look at our population, so this purple insect, these have to be insects because they have 6 walking legs. That's one of the characteristics of insects. This purple insect is immigrating into this I don't know what that is, mustard colored population. So when they come in, they bring in some new alleles potentially. They might also take some genetic diversity out of this other population. So both of those happen simultaneously. In a lot of cases in nature, we have two populations next to each other, and there's regular gene flow between them. Individuals move in both directions. If we look at the population of Illinois and Indiana, particularly up here in the northern part of the state, people move back and forth between the two states all the time. On the other hand, if we're thinking of the states, we also have a population of people in Alaska. People don't move back and forth between Illinois and Alaska all that frequently. It does happen, but there's less gene flow between Illinois and Alaska than there is between Illinois and Indiana. Okay, we're going to look at one more, and then I think that's all we're going to finish up today. So let's look at genetic drift, which is sometimes called random genetic drift, or it's sometimes called genetic sampling error. And if you've heard the term sampling error before, you've probably heard it in a statistics class. When we look at a statistics class, there's always a large group that we could look at. For instance, suppose we wanted to find out whether people in the United States are going to vote for Trump or Biden in the 2020 election. We're assuming it hasn't occurred at this point. We could interview some people and we could find out who they say they're going to vote for. Now, obviously, there's a problem. They might not tell us the truth. They might refuse to respond to our questions. But if we take a big enough sample, there are some things that we can do to compensate for those types of errors. And we can get a pretty accurate estimate of what the overall population results will be. But there is a problem. If we take a really small sample, suppose I want to figure out who people are going to vote for, Biden or Trump, and I only talk to 10 people. That's not enough to predict the outcome of the election. Why? Well, because with 10 people, I can't really get a good sampling of all the different racial, ethnic, religious, whatever categories you want to divide people into. I can't get a good sampling of all the different groups. So if I only talk to 10 people, I could get very misleading results. So genetic drift is similar, except in genetic drift, something happens in nature so that we start a population with a very small number of individuals. And since we start a population with a very small number of individuals, this means that when we do that, we might not get a representative sample from the overall population. So one type of genetic drift is something called the founder effect. In the founder effect, we have an original population shown over here on the left. We've got some red insects and some yellow insects. It looks like they might be ladybugs. Something happens. Maybe there's a strong wind and it blows some individuals from this population off of the mainland onto a small island. Depending on which individuals get transferred to that island, if all the individuals are the individuals with the alleles for this red color, then the population on this island could be completely red, even though the original population wasn't. And there really are ladybugs that are different colors besides red and orange. That's just kind of the stereotype. So think of if you ever saw that any reruns of that old TV series, Gilligan's Island. You have a bunch of castaways on this island. Well, if you look at those castaways, they're not very representative of the human population. There were no minorities on Gilligan's Island. If you ever saw any of the episodes, I have to think that even though one of the characters was called the professor, that they weren't really that smart a group. So I'm not sure they were representative of human intelligence either. So in the founder effect, one individual in this case is shown as starting a new population, but could be even a few small individuals. Think about kind of reaching into the grab bag that is all the people in the United States, 330 million people, and pulling out 8 or 10. Are you going to get a representative sample? Probably not. So if something like this happens, our new population has a very different allele frequency than the original population. And tomorrow we'll look at another type of genetic drift, and we'll talk about the other two major factors that change allele frequencies. And then we're going to work some Hardy-Weinberg problems and some natural selection problems. So as I say, make sure you have a calculator app handy, and you'll need it tomorrow. And one that will give you decimals also, obviously. Okay, any questions before we leave this page? If there are no questions,
No.
I'll stop screen sharing.
Friday,
I'm sorry, what?
like last Friday, you said there was an assignment due on Wednesday.
Mm-hmm.
Yeah, I couldn't find the assignment on D2L.
Since it was last week, it's posted under module 1. So it's due this week, but it's still posted at the end of module 1.
Okay.
Professor, I looked at the end of module 1 and I don't see it there either.
You don't see it there? Let me check and see if I can see it, because I looked earlier today and I thought I saw
don't see it either.
It shows on mine. It's after the Hardy-Weinberg PDF document. It's called monohybrid cross. Oh no. Now you can see it. It had it hidden, my bad. So this will be due tomorrow before we come to class. Should only take you about 5 minutes. This is, if you remember from 105, this is a monohybrid cross. So hopefully that still sounds vaguely familiar to you. Should be able to see it
I was
now.
going crazy.
No, the problem is this, when I create something, it automatically hides it so that you can't see it. That's the default. So I have to remember to show it to you. So that seems backwards to me, but oh well.
Yeah, that doesn't seem convenient.
Seems like, why would I post an assignment or give you a set of lecture notes if I didn't want you to see them? But I guess that's why I'm not a computer programmer.
Thank you.
Okay.
You're welcome.
Thank you. I can see it now.
Good. Because if you couldn't see it now, I'm starting to run out of ideas.
Okay. Have a good day.
You too. Bye-bye.
Have a good day.
Bye-bye. See you tomorrow, Jade.
Professor, I had a question.
Sure.
When you go back to the graphs for the types of selection, I understand that like the population, like the X and Y axis was like population and the other one was variance, I guess.
Yeah, the
Like, could you
x-axis and all
explain
those
the-
is whatever characteristic, and the y-axis is the frequency of the population that has it.
Oh, okay, the frequency of it. Because I was wondering, like, is that something that we really need to pay attention to be able to identify based off of the graphs?
You should be able to recognize it by the graph, and either tomorrow or Friday, depending on how far we get, we'll see some examples of that.
Okay. All right, well, that answers my question, though. Thank you.
Okay,
Have a good one.
you too. See you tomorrow, Matt.
For genetic drift, does that explain like the island gigantism at all?
It does in a way, but usually when we look at species on islands, a lot of times they're either bigger or smaller, and a lot of that's more usually a result of the fact that when a new type of organism gets to an island, there might not be predators around.
Okay.
And so it's really more kind of a relaxation of natural selection. You might have had to be small to crawl into a hole to avoid predators. But if you land on an island, man, there aren't any predators. For instance, a lot of the birds that can't fly, not ostriches, but things like dodo birds and things like that, actually evolved on islands where there weren't predators, so they didn't have to worry about being able to fly to get away.
Okay, it makes sense.
Size is kind of the same thing, you know. Hey, if you don't have any bigger animals around where you have to worry about getting noticed, all of a sudden there's a little more flexibility in your size. And on a lot of islands, we really see humans, we see not just humans, but we see animals get smaller just because there's a shortage of food. So a few years ago, you might have heard about the new kind of subspecies of humans that was discovered that was called hobbits, because they were only about 4 feet tall and they lived on an island.
No, they were just malnourished, basically.
Yeah, and we think over time they gradually evolved to become smaller because there weren't enough resources to have a very big population if everybody was 6 feet tall, you know. So one way to avoid starving is to be small. You don't need too many calories that way.
Okay, thanks. See you tomorrow.
Bye-bye. See you tomorrow, Nick.
Mr. Griffith?
Mm-hmm.
Yeah, so I don't know how we can get the videos like this one because I'm really lagging behind a little bit right now.
Okay.
Yeah, so I have to
Send
watch
me
it,
an
like,
email and
twice.
remind you, and I can send you. I'll send you this video as just as an attachment.
Not really, not only this, I mean like the last 2 classes, because I have to watch it like twice before I can get the
Okay.
whole of it well.
Yeah, if you send me that, remind me. I will see if I can send you all 3. It might be too big. I might have to send you a couple of emails.
Yeah.
You know,
that's
sometimes
okay.
there's a limit on how big attachments can be.
Yeah.
But once I get the Facebook page up, then you should be good, I think.
Okay. Thank you. I just want to get in touch with it before it start getting too much, you know.
Nope, I understand.
Yeah. Thank you.
You're welcome. Bye-bye.
Yeah, bye.